Hi, thank you so much for the invite. I'm so disappointed I can't be with you all in Montreal, but I'm expecting a baby any day, so I don't think it would be the smartest to get on a plane right now. So I'll get straight on with it because I know we're a little short on time. I'd like to talk to you today about a project that I was really excited to be a part of over the last couple years, mostly done by an extremely talented PhD student, Ariana Manardi, who's now doing her postdoc. And we explored using network control theory to guide TMS. So to guide brain stimulation. So the human connectome, I'm sure I don't have to motivate people in this crowd, but we've learned an awful lot about it in a very short amount of time. We know that the networks of the human brain contain a number of organizational features at the network level. These include rich clubs, they include an existence of modules, a balance between wiring cost and efficiency of the network. And what's especially powerful about this, to me at least, is that if we start to understand what a healthy brain looks like, we can understand a lot about what an unhealthy brain looks like. So for example, depression. Depression is one specific disease or disorder that can be viewed as a disconnectivity issue. People have been able to relate increases and decreases in connectivity within specific subnetworks, default mode, reward, attention, and cognitive control being shown here, to symptoms of depression, including rumination, anhedonia, dysphoria, and disrupted cognitive control. And especially interesting is when you are able to repair some of these network changes, say via medication, you start to see a reversal in these symptoms. You can link the symptoms to the network structure. So why we're all here at this symposium is to talk about stimulation. And of course, one treatment for treatment-resistant depression is transcranial magnetic stimulation. But what's done in clinical practice currently is that you literally just measure by fingers on the back of the head of where you should be stimulating, and assuming that this is the best site to stimulate for everyone. But here's the thing, we're all different. We're all really different. We're an incredibly diverse species, and this is both a wonderful thing and an incredibly challenging thing for those of us in neuroscience. In fact, we're so different that we can fingerprint our brains. You can turn up for a neuroimaging scan one day, come back a few weeks later and have a scan, and we will be able to tell it to you. Even between twins, we're able to tell the difference. So this is where I enter network control, just to give you a brief introduction of this. If you think about like the diagram you have in front of you here, this is a pulled apart car. There's lots of components that make a car, but when you're driving, you only need a few of them, specifically the steering wheel, the accelerator, the brake, and perhaps the clutch, to actually move this car, to drive it in places that you want to go, to control it. So network control comes in and says, okay, you give me that wiring diagram, you tell me how these components are connected. Could I tell you which part is the steering wheel? Could I tell you which part is the accelerator? And this builds on a mathematical framework that's been around for many decades at this point, called control theory. And it all comes down to, like, the first theorem in control theory is saying that a system is controllable if we can drive it from any initial state to any desired final state in finite time. And if that's not what we're wanting to be doing in the brain, it's obviously a great map. So for the following, we adopt a linear time invariant dynamics to describe open loop control, and this has a neat mapping to transcranial magnetic stimulation. So in this dynamical equation, X is telling us the dynamics of nodes in the brain, so each node is a brain region. A is the adjacency matrix, so this is the connectome. And then B is telling us where we're going to apply this stimulation, where we're going to apply the TMS signal. And U is going to tell us the nature of this stimuli. And then with this framework, it allows us to think of TMS not just as not just as a quick perturbation in the same place for everyone, but we can start to think of it as a network intervention. We can look at everyone's individual connectome, and we can start plotting out what might happen if we perturb that network at different places, or in different magnitudes or different styles. And we can think of our brain as moving through this activity space, where some parts of this landscape, some of them will be unhealthy, say associated with depression, some of them will be healthy, but we all have our own individual landscapes, and we can model how to navigate these landscapes using network control theory. So what we did right from the very beginning was make everything as individual as possible. So that means individual anatomy, individual parcellation, individual structural connectome, and individual graph construction. We then overlaid this with knowledge of some common resting state networks. We used the breakdown from Yao et al. 2011 into 7 resting state networks: default mode, frontal parietal, visual, limbic, somatomotor, ventral attention, and dorsal attention. And what this gave us was kind of the following. So these are abstracted graphs of just 3 example subjects. They do represent their brains, but they're more in a topological space, which is why they don't look like brains. But what I want you to really take from them is, just at a high level, they look the same. You've got 2 clumps. These are each of the hemispheres, the left and the right. The yellow dots, they're the cerebellum, and they're somewhat less well connected with the rest of it. So you're seeing this same kind of qualitative structure, but when you look more at the details, you'll actually notice a lot of differences. There's a lot of differences in the connectivity of each particular node, the connectivity within each particular network, and this is all embedded in these individual connectomes. So then what we set out to model was, okay, I've got my seven resting state networks. What if I want to activate one of them? I want to activate all of the regions that are associated, in this example, to the somatomotor network. So we used structural data, DTI, to create the connectomes, the wiring diagram to run the simulation on. We then coupled it at this point with functional fMRI data to give us real activity states. So our baseline state is taken from the real data. Our target state that we want to drive the system to would be an activated resting state network. And the way we get there is by simulating a TMS pulse and applying it to that baseline state to drive it to the final state. Now, how do we choose where to stimulate? We had 3 different scenarios. The first one that we're calling traditional sites, this is what's found in the literature. This is the current clinical practice. And importantly, these nodes are the same for everyone. Every single person gets stimulated in the same place. The second thing we did was the most simple network-based approach that we could come up with that was reasonable. So this was based on path length. So we identified the regions that had the smallest topological distance to the network of interest. So we identified the nodes that had many connections to this specific resting state network. And now this is personalized. This is going to be allowed to vary per person. And then finally, we have the network control theory sites. So these are the regions that when we run the simulations, the open loop control simulations, they predict which nodes will be activated the most in the network of interest. This will also be personalized. Another comment, the current state of the art allows us to stimulate 2 nodes at once. And computationally, we can do whatever we like. So we brought in, to match the clinical standard, we brought in that we can stimulate 2 at once. So we can pick the best 2 every time to get the maximum effect. We compared to two different null models because there's two other driving factors that really may affect things. One is topology of the network, and the other is space. If you're closer physically to a particular region, then the effect of a TMS perturbation is likely going to have a bigger effect just because you're closer. So we have a topological null model where we shuffle the edges, but we preserve degree, strength, and weight distributions. And we have a spatial null model that preserves the weight-distance relationship that we see in spatial brain networks. So we go ahead and we try these different stimulation scenarios, and all of them are going to get us close, right? But we want to know which one does best. And because of the nature of this simulation, the control theory ones, we know they're going to come out the best because they describe the dynamics. But if the traditional, the clinical standard was good enough, they should be pretty close. And if we didn't need something as complex as control theory, the path length should also be close. So let's see what we find. In this example, I focus on default mode network. So you've got the 3 rows looking at traditional path length control theory, and they do all reasonably successfully activate most of the default mode. But importantly, the control theory stimulation sites, it gets there faster and it covers more of the network. It does so especially via some temporal regions that are really poorly reached by the other 2 methods. We go ahead and count the number of nodes that are stimulated within the top 30 that are activated within the network, and you see this increasing, getting better. So clinical standard is the orange and red. The path length is then blue, so we're seeing a little bit of an improvement, and then control theory is the green. We're seeing a big improvement, significantly more so than clinical practice. The gray ones are the null models, so it's showing they have a contribution, but really not significant on the rest of it. So the next question is, how specific is it? It's all well and good saying I've activated the default mode network, but what if I just activated everything? Then I can still claim a win. Well, it turns out we were actually able to be pretty specific, a specificity of about 96% in the case of default mode network. Coming back to individual variability, because I was really stressing at the beginning how different our brains are. So if we've identified which nodes via network control theory are best to activate the brain, where are they, and which connections are they most connected to? So this is an example of subject 1. The yellow node is the one we identified as the optimal place to target, and that's just in one brain. If we overlay everyone, it looks like this. So the bigger the node, the more people it appears in. So you do see some overlap, but that's a huge amount of variability to target one resting state network. I will note here that, of course, not all of these are physically tractable to go ahead and stimulate, but we can cut those out and recommend the next best ones from a computational point of view. And from just literally choosing which are optimal, these are the ones. A quick aside of why this is important, from a really cool paper from some of my colleagues at Northeastern, they had a group of people suffering with schizophrenia, and they taught them mindfulness meditation. And with some feedback in the fMRI scanner, they were actually able to suppress the activity in their default mode network. And following this practice, they actually experienced a reduction in auditory and visual hallucinations for several weeks following the practice. So this can have a big effect on people's lives. Circling back to depression, one of the motivators at the beginning of this talk, we extracted the regions that were most associated with depression via neurosynth. So just a meta-analytic study of looking at all papers that are out there on depression. And we ran the same kind of analysis to identify regions that can stimulate those associated with depression. And we found the same results. The network control theory, again and again, outperforms clinical standard. So in summary, our simulations predict that the network control framework can provide a way to systematically assess the efficacy of stimulating different target sites to activate whole subnetworks. We predict that network control can identify target sites that greatly outperform other methods, and even simple path length approaches from network science perform better than current clinical standard. So it's really critical that we take the individual network into account. Physical proximity is not enough, nor is the group average. We hypothesize that trajectories induced by network control predictions will be more direct and require less control energy, but we're still looking into this. And this has potential applications for disorders including depression and for improving cognitive function. P.S. I have no disclosures. And whoops, sorry, I just nearly skipped this slide. Of course, thank you to the team that it took a village to do this paper. And I especially again want to shout out to Ariana, who carried the bulk of this work here. So yeah, thanks. I'm afraid I can't take questions because I'm not there, but please email me. My details are on the screen. You can use my uCalgary or Gmail address and find me online. I would love to hear from you. So thanks very much.